Hi, and welcome to this Skintellectual webinar on retinol, the basics of retinol treatment. Retinoids are the derivatives of vitamin A acid, which can bind, vitamin A, which can bind and activate specific nuclear receptors and induce transcription of certain genes in the cell. The discovery of retinol structure was done by P. Kerr in 1931 and was awarded by a Nobel Prize. The first milestone in the research of retinoid tretinoin in skin aging was published in 1983 by Albert Kligman and colleagues. There are 4 generations of retinoids. The newer generations are specific and more targeting acne, and they are prescription-only preparations. Active form of retinoids in the skin is trans-retinoic acid. In order to be transformed to trans-retinoic acid, retinol should pass through a 2-step oxidation process in the skin. Derivatives of retinol should pass more steps of oxidation and conversion. Retinoids are hormone-like cosmetic ingredients with genomic and non-genomic action. The genomic activity is the most important one for restructuring of the skin induced by retinoids. Retinoids bind specific proteins, transporting them inside the cell and all the way into the nucleus of the cell. Inside the nucleus, retinoids bind specific promoter genes and thus modifying DNA transcription and therefore messenger RNA and protein synthesis in the cell. The anti-aging effect of retinoids relates to the changes of, first, on a molecular level, increase of collagen synthesis, dermal and epidermal proliferation, and inhibition of tyrosinase. Second, the structural level, the skin morphology restructuring, which means thickening of the dermis and the epidermis. There's an increase in collagen deposition, a normalization of elastin organization, a decrease in the melanin content, and neoangiogenesis, formation of new blood vessels. And third, finally, clinical effects, which we see in improvement in wrinkling, sagging, skin texture, and discoloration. As an illustration of hormone-like effects of retinol, this biopsy study demonstrated significant skin restructuring on the sun-protected skin of 12 females, which were 76-year-old on average. And only 7 days after starting of topical retinol cream, there was the effect. Analysis of buttock skin biopsies after retinol treatment showed an increase in dermal and epidermal cells, growth of dermal vascularization, and increased expression of collagen type 1 and collagen synthesis markers. Therefore, retinoid tretinoin is the only approved topical treatment of photoaging by the FDA in the USA. Then, side effects. The side effects of tretinoin limits its use in cosmetic practice. The most typical side effects are skin dryness, scaling, and irritation, also giving some redness, in some cases to the level of even dermatitis. So that it even has a name, it's called retinoid dermatitis in dermatology. Can retinol as a safer option replace tretinoin in photoaging treatment? The answer is yes. In the presented double-blind split-phase study of 0.25, 0.5 and 1% retinol were therapeutically equivalent to 0.025, 0.05 and 0.1% of tretinoin in the clinical reduction of photoaging. And the clinical reduction was assessed by blinded evaluators. Meantime, retinol demonstrated a better safety profile compared to tretinoin, as it's presented in the review of these 3 studies that you see here. Retinol showed minimal irritation levels, and which means signs of erythema and irritation, and induced less dryness of the skin and less scaling, typical for tretinoin. So that concludes our webinar on the basis of retinol treatment in skintellectual solutions. Thanks for watching.